Bismillahirrahmanirrahim. My dear students of second year MBBS, today I am going to talk about the glands which are associated with the gastrointestinal tract, and the salivary glands are one of those very important glands. Dear students, आज काफ़ी अरसे के बाद आपसे मुखातिब हुआ हूँ। 15 रोज़ की समर वेकेशंस के बाद दोबारा ऑनलाइन क्लासेस शुरू करते हैं। अगरचे ये एक बहुत बदमज़ा सी कोशिश है, आप सामने हों तो पढ़ाने का लुत्फ़ दुबाला हो जाता है, लेकिन क्या करें, कोरोना की वजह से। वीरां है मैं कदा खुमो सागर उदास हैं, तुम क्या गए कि रूठ गए दिन बहार के। मेरी दुआ है कि अल्लाह इस कोरोना की मुसीबत से हम सबको निजात अता फ़रमाएँ और हम बहुत जल्द दोबारा मिलें, दू बदू और रूबरू एक दूसरे के सामने, और फिर पढ़ाई का मज़ा नए सिरे से आ। Let us start with the revision of the classification of the glands. I have told you in first year MBBS there are different types of glands, and before talking about the salivary glands, let's revise our previous knowledge of the classification of the glands. There are two types of glands, the exocrine and the endocrine glands. As you know, the endocrine glands are the glands of internal secretion, and these glands, they pour their secretions directly into the bloodstream without a duct system. And exocrine glands are the glands of external secretion. They secrete their products through a duct. The gland in this group can be divided into three groups. There are merocrine glands, the apocrine glands, and the holocrine gland. I have told you in first year MBBS that the merocrine glands are the glands which secrete, merely the secretion is poured out without any cellular component, while the apocrine glands, a portion of the secreting cell's body, for example the apex of the cell, apical cytoplasm, is also extruded out. And holocrine, as the name indicates, the whole of the cell, the entire cell disintegrates to secrete its substance. The sebaceous glands are the examples of the holocrine type of gland. A type of secretory product of an exocrine gland may also be one of the three categories. There are serous, mucous, and mixed. Serous secretion is watery, often protein rich. It is enzymatic, while the mucous glands secrete a viscous product which is rich in carbohydrates, that is the glycoproteins. Salivary glands are compound tubulo-alveolar glands of merocrine variety. These are exocrine glands. The ducts of these glands open into the oral cavity. Oral cavity is kept moist by a film of fluid called saliva that coats the teeth and the mucosa. Saliva is a complex fluid produced by the salivary glands. Classification of the salivary glands. If we talk about the salivary glands, there are major salivary glands, which are three pairs of glands, the parotid, submandibular, and sublingual, located outside the oral cavity. While there are minor salivary glands, these minor salivary glands are located in various parts of the oral cavity. They are the labial, the lingual, the palatal, the buccal, the glossopalatine, and retromolar. These are very small glands. Based on the type of the secretion, we have got the serous type of. As regards the serous glands, the parotid gland is the serous type of gland. Parotid gland is purely a serous type of gland. The sublingual is mucous. The sublingual and submandibular glands are actually the mixed glands, but the submandibular is predominantly, it is a serous type of gland, and the sublingual is predominantly, it is a mucous type of gland. You can see this is the parotid gland, parotid gland lying in front of the auricle or pinna. If we talk about the general structure of the salivary glands, the major salivary glands consists of the parenchyma, stroma and the duct system. The parenchyma, as I have told you, the actual functional portion is known as the parenchyma, and I have told you that these glands are serous as well as these glands are mucous type of glands. So when we cut a section, we will find the serous or mucous alveoli. The serous and mucous alveolus is it is a dilated sac-like structure, and because these are compound tubulo-alveolar, so there are alveoli. When we cut the section, we will find a lot of structures which are having the pyramidal cells, and these cells are surrounding a lumen, and these pyramidal cells are serous cells and mucous cells, depending upon which gland we are going to study. The parenchyma is composed of the alveoli, but because some portions of the ducts are also involved in the secretory process, the major salivary glands are included in the tubulo-alveolar variety. Serous cells. Serous cells, the morphology of the serous cells is, these cells are pyramidal in shape, they have got a broad base, they are lying over the basement membrane, and the apex is towards the lumen. The nucleus is spherical and placed at the basal region. The basal cytoplasm is packed with parallelly stacked rough endoplasmic reticulum, which is placed basal and lateral to the nucleus. The lumen usually has finger-like extensions located between the adjacent cells called intercellular canaliculi. This is a serous type of cell. You can see in the serous type of cell there are zymogen granules. This is zymogen cell. So the upper portion of this type of cell is eosinophilic because of the presence of these zymogen granules, but its lower portion, because the nucleus is present in the basal half of the pyramidal cell, and just beneath the nucleus there is a lot of rough endoplasmic reticulum which gives basophilia to this type of a cell. There is a very well developed supranuclear Golgi body, and these cells are present above the basement membrane, and the cell is opening into the lumen of the alveolus. These zymogen granules are the glycolated proteins stored in a vacuole. Apical cytoplasm shows secretory granules 1 millimeter in diameter. The granules are closely opposed but retain their individuality. The mucous cells. Its structure differs from the serous cells. The apex of the cell appears empty except for thin strands of cytoplasm forming a trabecular network. Nucleus and the rims of cytoplasm are compressed against the base of the cell because this cell has got it is a goblet-like cell. The goblet cells, I have told you, it is a unicellular gland and the mucus secreting gland. And mucinogen granules, which are actually, they secrete the glycoprotein which is hydrophilic, which when comes out and it then combines with water to gives rise to a foamy gel-like material which is called the mucus. And when we see under the microscope, these cells, they are empty towards the apical site. Nucleus and the rim of the cytoplasm are compressed against the base of the cell. Nucleus of the mucous cell is oval or flattened in shape and located just above the basal plasma membrane. Mucous cells show accumulation of large amount of secretory product. These are the mucous cells. You can see this is the elongated nucleus which is towards the basement membrane, and there are a lot of mucinogen granules which are present towards the apical site of the cytoplasm. Mucous cell is seen to be filled with pale, translucent secretory droplets containing scattered flocculent material, unstained in routine histological sections. Special stains like PAS and Alcian blue, it is heavily stained. Mucous secretion differs from the serous secretion in two ways. They have little or no enzymatic activity and mainly serves for lubrication and protection of the oral tissues. Number 2, the ratio of carbohydrate to protein is greater and large amount of sialic acid and occasionally sulfated sugars is present in. Can you see these are the mucous alveoli? These are the mucous alveoli, and I have told you that the sublingual gland is predominantly it is a mucous type of gland, and in a mucous type of gland you will see these type of alveoli. These alveoli are they have got a very prominent lumen. You can see the lumen is visible. The cell walls and boundaries are clearly distinguishable. The cells are lightly stained pinkish in color, and the nuclei are present around the periphery. The nuclei are just above the basal lamina. These are flattened and elongated nuclei, and they are present just above the basal lamina because these nuclei are pushed by the heavy secretion. The mucous secretion is a heavy secretion, and it pushes the cytoplasm, the rest of the organelles and the nucleus towards the basement membrane. So this is the mucous type of gland. Here you can see that I have told you that the parenchyma of the glands, the salivary glands, it is composed of the alveoli and the duct system. And the ducts are of three types. There are intercalated ducts, there are striated ducts, and there are secretory ducts. You can see these are the alveoli, and these alveoli, when we cut a section, these are the serous cells, these are the mucous cells. Another very important feature of the parotid gland and the sublingual and submandibular gland is that around these pyramidal cells we have got the star-shaped cells, which are modified epithelial cells. They are myoepithelial cells. They are contractile cells associated with the secretory end pieces and intercalated ducts of the salivary glands. Myoepithelial cells are similar to smooth muscle cells but are derived from the epithelium. But in the cytoplasm of these cells there are actin filaments and the dense bodies just like the smooth muscles. These cells are located between the basal lamina and secretory or ductal cells. The plasma membrane of the myoepithelial cells joins the basal membrane of parenchymal cells by desmosomes. So these are star-shaped cells having cytoplasmic processes, and they push the secretion. When they contract, they push the secretions into the lumen. So these are the cells which facilitate the secretion of the saliva when need arises. These are stellate or spider-like cells with flattened nucleus and flat perinuclear cytoplasm and long branching processes that embrace secretory and duct cells. Their appearance is like basket cradling the secretory unit, so-called basket cells. The cell organelles are restricted to perinuclear cytoplasm. Their structure is similar to smooth muscles. Immunofluorescence studies indicate presence of myosin actin. There is regular pulsatile movement of the entire unit. Difficult to identify under the light microscope, can be identified by immunohistochemical or immunofluorescent techniques. Contain cytokeratin intermediate filaments and contractile actin filaments. This helps to identify their immunohistochemically. Functions: they accelerate the initial outflow of the saliva from the acini. contribute to secretory pressure in the acini or duct, supporting the underlying parenchyma and reduce the back. Let's talk about the ducts, intercalated ducts and the striated ducts and the excretory ducts. Intercalated ducts are the smallest ducts. They are known as the intralobular because they are present in the. Here, I must tell you that these tubuloalveolar glands, they are surrounded by the connective tissue capsule, and these connective tissue capsules, they send septa or trabeculae inside to divide the gland into smaller compartments, which are the lobes and lobules. There are septa and septula, which gives rise to the lobes and lobules, and the smallest portion, the lobule, it contains the acini and intralobular ducts, and then we have got the striated ducts and then interlobular ducts, which are excretory ducts. Intercalated ducts are lined by single layer of cuboidal cells with a relatively clear appearing cytoplasm. In electron microscope studies, the intercalated duct cells resemble the serous cells. You can see this is a very fascinating cell. Functions: they modify the saliva through secretory and resorptive process. They contribute to substances like lactoferrin. Intercalated ducts also houses undifferentiated cells which can undergo differentiation and replace the damaged cells in the end pieces of the striated ducts. As regards the striated ducts, these are lined by tall columnar epithelial cells with centrally placed nuclei. The cytoplasm is eosinophilic and show prominent striations at the basal end of the cells, perpendicular to basal surface. That's the reason we call these the striated cells and striated ducts. Electron microscopic studies revealed that the basal cytoplasm of the striated duct cells show deep infoldings of the plasma membrane, and many large mitochondria, usually radially oriented, are located between the infoldings, indicating that the cell is involved in active transport. Can you see, this is the cell which is known as the striated cell. You can see towards the basal side there are infoldings of the cell membrane, and these infoldings of the cell membrane contain elongated mitochondria. Functions of this type of a duct: these are site of absorption of sodium chloride and excretion of potassium and bicarbonate. The reabsorption is against the concentration gradient, so needs more energy. That's why a lot of mitochondria are present. Excretory ducts: as the excretory duct enlarges, it contains two layers, the mucous and connective tissue adventitia. The mucosal epithelium is of pseudostratified columnar ciliated variety. Occasionally there are goblet cells and ciliated cells are seen. The ductal epithelium slowly undergoes transformation to cuboidal and finally to stratified squamous epithelium. Connective tissue elements, that is the stroma of the glands. The cells include the fibroblast, macrophages, mast cells, occasionally leukocytes, fat cells and plasma cells. The connective tissue forms the capsule for the gland and divides the gland into lobules. The vascular supply of the gland is also embedded within the connective tissue. Minor salivary glands: these are located beneath the epithelium in almost all parts of the oral cavity. They open by short duct directly into the mouth. They lack distinct capsule and instead mixing with the connective tissue mucosa. Salivary glands are absent in anterolateral part of the hard palate and the gingiva. Saliva is the secretion of major and minor salivary glands along with desquamated epithelial cells, microorganisms, food debris, serum components and inflammatory cells. Each gland's secretions differ in amount and content. Parotid gland, watery saliva, enzymatic, amylase, a lot of amylase is present in the saliva of the parotid gland. Proline-rich proteins and glycoproteins. The submandibular gland, in addition to the above glycosylated substances, that mucins are present in the sublingual gland, viscous saliva rich and mucin. Composition of the saliva, daily we have got 600 to 100 milliliter. There are sodium, potassium, chlorides, bicarbonates, calcium, magnesium. The secretory proteins are the amylase, proline-rich proteins, mucins. There are immunoglobulins present, the secretory immunoglobulin A, IgG and IgM. The small organic substances, the glucose, amino acid, urea, uric acid and lipid molecules are also present. Other components of the saliva are the epidermal growth factor, insulin, cyclic adenosine monophosphate. Parotid gland is the largest salivary gland. Its parenchyma consists of only serous alveoli and its secretion is purely serous in nature. The intercalated ducts are long and therefore prominent in sections. The parotid gland is the largest salivary gland. Its parenchyma consists of only serous alveoli and hence its secretion is purely serous in nature. The intercalated ducts are long and therefore prominent in sections. The connective tissue capsule is thick and well developed. The secretory product has a high amylase activity, I have said. This is the sublingual gland, and the sublingual gland, this is also a mixed gland, predominantly mucous in type. The parenchyma consists of mucous alveoli, some of which bear serous demilunes. Serous demilunes are the caps of the serous type of cells present on one side of the mucous alveoli. The pure serous acini are rarely seen. Both intercalated and striated ducts are short and thus less prominent in sections. The connective tissue capsule is thin and poorly developed. This is the submandibular gland. The submandibular gland belongs to the mixed variety of exocrine gland, but it is predominantly serous type of gland. Its parenchyma consists of serous and mucous alveoli. Some of the mucous acini bear caps of serous cells called serous demilunes. Demilunes from semilunar, that is crescent-like, which give a crescent-shaped appearance in sections. The serous cells of the demilunes pour their secretion into the lumen of the alveolus through intercellular canaliculi. So in between the cells there are canaliculi seen in this case. The demilune cells produce lysozyme. In the submandibular gland, the intercalated ducts are narrow and short, while the striated ducts are wider and longer. The connective tissue capsule is well developed. The saliva from this gland has a low amylase activity and contains lysozyme which breaks down the wall of the certain bacteria. Let's talk about the functions of the saliva. As you can see in this table, functions of the saliva include the lubrication, digestion and protection. The saliva constantly moistens the oral cavity and acts as a lubricant during mastication, deglutition and speech. The saliva contains the amylase that initiates the digestion of the polysaccharides. in the buccal cavity. The oral cavity is loaded with pathogenic bacteria that can destroy tissues and can also cause dental caries. So the saliva protects the oral tissues against the harmful effect of these microbes. This important function is carried out in the following two ways. The flow of saliva washes away the pathogenic bacteria as well as the food particles that provide their metabolic support. The saliva contains several factors which actually destroy the bacteria. These factors include the lysozyme, thiocyanate, and the IgA.